Hello everyone, this is the post lab for exercise 5 entitled Membrane Transport and Donan Equilibrium. I am Kimberly M. MASAMAYOR.
And I am John Telen, and we are Group 5.
In keeping the cell alive and metabolic processes going, the cell must maintain concentrations of certain substances. The title of this exercise is Membrane Transport and Donan Equilibrium. This is all about the principles of transport of these substances in and out of the cell. The semi-permeable membrane of the cell serves the dynamic function of regulating the flow materials in and out of the cell. This membrane is composed of two phospholipid bilayers with numerous proteins either embedded or loosely attached to their surface. Hence, S. J. Singer and G. L. Nicholson proposed their fluid mosaic model of the cell membrane as shown in the picture. It has different kinds of protein channels, and carbohydrates. Carbohydrates in the form of oligosaccharides may also be attached to the lipids forming glycolipids or to the proteins forming glycoproteins. They form hydrogen bonds with the water molecules surrounding the cell and thus help to stabilize membrane structure. As you can see, it has an asymmetric nature where the phospholipids and proteins are asymmetrically distributed between inner and outer leaflets of the membrane and the flip-flop to each other in either side of the membrane. Therefore, cell membranes must maintain an appropriate asymmetry of phospholipids, proteins, and other molecules to facilitate and display receptors, signaling molecules, and other structures for efficient function of the cell. Molecules will eventually diffuse across the protein-free lipid bilayer down its concentration gradient. The diffusion rate depends on the size and the solubility of this molecule in oil. The smaller and the more hydrophobic the molecule is, the faster it will diffuse to the lipid bilayer. However, for the charged molecules, the lipid bilayer is highly impermeable because the charge and the hydration of ions prevent them from passing the lipid layer. Therefore, it requires these channel proteins. Now, since we are talking about the membrane transport, we will discuss the two types of transport across the cell membrane, and these are the passive transport and the active transport. Passive transport does not require energy, thus ions can move freely across the semi-permeable membrane. This is because in this kind of transport, materials can be transported along their electrochemical gradients, and ions move with the electrochemical and concentration gradients moving from high to low concentration and a high to low overall charge. It may be directly through the lipid layer or via channel or carrier proteins. In diffusions, ion can fit to the opening or holes in the membrane, while in facilitated diffusions, ions move across membrane protein. On the other hand, active transport entails the movement of the substances against their electrochemical gradients. It requires energy which can be provided by ATP, light, or by energy stored in an ion gradient. According to Alberts et al., ATP-driven active transport are inhibited by metabolic inhibitors of ATP production. An example of ATP-driven active transport is the sodium-potassium pump, which is crucial in the maintenance of the membrane potential of cells. The ions here go against the concentration and electrochemical gradients moving from low to high. Ions are vitally important to the metabolic reactions, and they are charged and insoluble in oil, which means they cannot pass directly to the lipid bilayer, and their diffusion is affected by two types of gradients. Again, the concentration and electrical gradients. Now let's talk about what is the Donan equilibrium. It was named after Frederick George Donan, who demonstrated the effects of the two gradients. It was observed that there is a large concentration of non-diffusible anions within the cell in the form of negatively charged proteins, which creates an electric gradient that prevents or lessens the entry of anions while favoring the entry of cation. While the electrical gradient is operating, the concentration gradient also continues to influence ion diffusion. The cation would be pulled in by both concentration and electrical gradient, and the anion will be pulled by the concentration gradient and repelled by the electrical gradients if a high concentration of certain cation and anion is placed outside the cell. This continues until an equilibrium is achieved where both sides of the membranes become electrically neutral. This is what we call Donan equilibrium or Gibbs Donan equilibrium. In amphibian skin, the direction of the movements of ions in the Na plus and K plus pump is opposite in frog or toad skin. Here, the exterior surface is specialized to transport Na from the pond water to the interstitial fluids at the inner tissue called the cryon layer. By this mechanism, the frog can trap its essential iron for the use in different tissues. K plus is pumped out, but much more Na plus is pumped than the K plus pumped out as shown in the figure. Thus, the inside surface of the skin becomes relatively negative compared to the outside. So for the objectives of this study, at the end of this laboratory exercise, we should be able to differentiate passive and active transport, discuss the principles of Donan equilibrium and active transport using simple experimental models, and determine the effect of cyanide, a metabolic inhibitor, on active transport. So for the materials, these are the materials used along with the biological sample, solution, and equipment and labwares. For the methods, this is the flow chart for the demonstration of Donan equilibrium. Well, this is the flow chart for the demonstration of active transport. The flow chart for the effect of cyanide in active transport, and the calculation of active transport rate along with the formula. The rationale for each of the methods will be explained in the study questions. Since we cannot perform this exercise in Mall Bio Lab, a video demo was provided and the data were given for analysis.
For the results and discussions of the exercise, these are the results derived from the methods that are done for the exercise. For the demonstration of the equilibrium, table 5.1 entails the average pH readings of solution outside the bag for 5 replicas over 90 minutes, and table 5.2 indicates the average pH readings of the solution inside the dialysis bag for 5 replicas over 90 minutes. We can observe that there is a equilibrium happening at the 90th minute, wherein the final pH of both the solution inside the dialysis bag and the solution outside the bag has an equal pH of 3.2. We can say that there is a fluctuating movement in the H plus ions, in which these H plus ions are stimulated by the gelatin added in the solution. The accumulation of H plus ions from outside to inside due to electric and concentration gradients account for the change in the pH inside the solution inside the dialysis bag and the solution outside the dialysis bag. For the demonstration of active transport, table 5.3 demonstrates the average weight and active transport rates of toad skin bag in linear solution over 90 minutes. The bag loses weight up until the end of the 90th minute, although fluctuates at the 45th minute. The partial rates also constantly decrease aside from the temporary increase at the 45th minute. The negative rate of active transport meant that the bag has lost some weight due to the movement of ions. So as we can observe that the final weight of the bag means it has lost some ions. It has lost some ions manifested by the value of the average active transport rate, which is negative 7.857 times 10 to the negative 0.5. Next is the effect of the cyanide on active transport. Table 5.4 demonstrates the average weight and active transport rates of toad skin bag in linear solution with cyanide over 90 minutes. So we can see that as compared to the normal setup, the bag loses weight less than what is compared to the normal setup. The partial rates and the average weight can also be seen to be lesser than that of the normal setup without cyanide. Therefore, we can say that the cyanide inhibits the rate of active transport as shown with the results. Inhibition means that it slows down the rate of active transport. Therefore, we can see that there is a less average rate of active transport in the cyanide setup compared to that without cyanide. For the study questions, for the first study question, the answer is that an artificial membrane was used in order to recreate a semi-permeable membrane. Therefore, we need to use the artificial membrane in order to simulate Donan equilibrium, since Donan equilibrium or cellular transport happens in semi-permeable membrane. Artificial membranes provide that semi-permeable membrane in order for specific ions to pass through. For the second question, we can observe that within the data that there is a behavior of the pH wherein the pH level of the surrounding acidified distilled water tends to fluctuate as it decreases and increases as time goes by. Although we can see that the final pH of the dialysis bag decreased, which manifested that the H plus concentration inside the dialysis bag increased, the observed behavior of the pH of the surrounding acidified distilled water is that as time goes by, it continually goes up and down with no consistency of increase or decrease observed over time. With this, we can say that we can observe the equilibrium as molecules move in regards to both its concentration and electrical gradients, and thus makes the molecules move back and forth up until the systems would reach an equilibrium in both electrical and concentration gradients, and also in pH. Therefore, we can observe such behavior in the experiment due to the equilibrium. For the third study question, an appropriate alternative would be using universal pH paper. Universal pH paper involves dyes in the paper wherein it changes according to the pH level. Then the colors would then be compared to a meter wherein this would indicate what pH the solution would be. Although the increments of the pH of the universal pH paper would be by 1, this means that it is a less accurate measurement compared to that of the actual pH meter. Although this would be a sufficient alternative for the pH meter in its absence. For the fourth study question, a low-cost experiment would be using a dialysis bag as a semi-permeable membrane and using diluted soy sauce as a medium. This would be a cost-effective solution in this demonstrating the equilibrium, as color change would be more manifested in this setup, as we could not rely for a measurement of pH since we do not have the materials for it. For the fifth study question, the purpose of the Ringer solution was to provide for sodium ions or Na plus ions and potassium ions to demonstrate the mechanism of transport of the sodium-potassium pump of the toad skin bag. The Ringer solution contains salts in which some of these salts are potassium chloride and sodium chloride. And as we know, salts dissociate when dissolved in water. Therefore, when potassium chloride and sodium chloride are dissolved in water, these are dissociated into Na plus ions and K plus ions, and also chlorine ions. Therefore, in order to demonstrate the active transport or the sodium-potassium pump in the toad skin bag, we need the Ringer solution to provide the sodium ions and potassium ions that would transport and move outside and inside the toad skin bag. Discuss the details of the mechanism of the sodium-potassium transport. For the sixth question, this is the mechanism of the sodium-potassium pump, or the sodium-potassium ions are transported in and out of the cell, as illustrated in Figure 1. Na plus ions attach to channel proteins embedded in the cell membrane of the cells and are released towards outside of the cell when the channel proteins, sodium ions, and phosphorylation is stimulated by energy in the form of ATP. Therefore, ATP is important in order for active transport and the sodium-potassium pump to occur. Therefore, in summary, the sodium-potassium pump moves against the concentration and electrochemical gradients and the surroundings, as active transport is utilized to move sodium ions outside the cell and potassium ions towards inside the cell. The next study question is this, and the answer is, when channel proteins in toad skins are completely different for the conventional sodium-potassium pump, as the toad skin moves potassium ions inwards to the cell and takes sodium ions outside the cell, channel proteins in epithelial cell membranes were completely different, as potassium ions binding to the channel proteins from outside the cell are moved inside the cell through active transport by energy in the form of ATP. Vice versa happens to that of sodium ions, which are pushed out of the cell. But still, considering that more sodium ions are taken in, then sodium ions are taken out, then potassium ions taken in. Channel proteins in the toad skin's epithelial cell membranes are much more different, which is manifested by the behavior of transport of sodium and potassium ions. If the skin bag is turned proper side out, will this difference in the side of the skin exposed to the medium alter the results? How and why? Yes, this will alter the results, since we can say that this will alter also the position of the channel proteins embedded in the membrane. Thus, this will also change the direction of the ions and the movement of the ions present in the medium and moving inside and out of the toad skin bag. Explain the change in the weight of the bag in the presence of sodium cyanide and discuss how cyanide affects the active transport. The changes in weight was far more lesser with the introduction of sodium cyanide, as dissociated cyanide ions from NaCN are inhibitors for mitochondrial energy production in cells. Cyanide inhibits the production of ATP in cells, and by principle, active transport requires ATP to occur. As active transport uses ATP to excite the channel proteins to either release molecules outside the cell or allow cells to enter the cell against both the electrochemical and concentration gradients. Thus, the lack of ATP production induced by the inhibition by the cyanide ions lead to a lesser movement of both Na plus and K plus ions inside and outside the skin bag, thus explaining the lesser change in weight. For the last study question, our answer is that the partial rates of active transport are far more or less in the setup where cyanide was introduced than that of where cyanide was not introduced, which had a greater partial and average rate of active rate of transport. Compared to the average active transport rate of the setup of cyanide, which is negative 3.29 times 10 to the negative 5 grams per millimeter squared, the average active transport rate is greater in the normal setup with an average active transport rate of negative 7.85 times 10 to the negative 5 grams per meter times millimeter squared. This means that sodium and potassium ions move more in the setup than cyanide, as cyanide is known to inhibit the production of ATP in the mitochondria of cells. This inhibition prevents the stimulation of channel proteins embedded in the membranes of these dermal cells, thus exhibiting a far more lesser rate of active transport. For our reference cited, this is the references that we have used. So that is all, and thank you for listening.